So welcome again to Pharmalytical Summit. Today I'm going to talk about powder XRD for pharmaceutical. I would like to acknowledge Dr. Mesij Garza, my Rigaku Europe colleague. The outline of my talk today is R&D processes of pharmaceutical drugs, investigation of crystal forms, quality check of API powder, formulation, Rigaku solution for pharmaceutical, and the summary. Let me also bring up the laser pointer. So in my view, I'm not a specialist in drug design, but these are four major processes in the drug formulation in the process of manufacturing of the drugs: physical and chemical approach, pre-formulation, investigational drug formulation design, and the products, including the scale-up. The powder diffraction is involved in each of these steps, and hopefully in my today's talk, I will try to show you that. Structure analysis of the target protein and API, which is included in the investigation of the compound. How many forms are there? The polycrystalline forms or the API, which is the best form of the drug, which is included in the investigation of the crystal forms. And then quality check of API powder, check of quality of API, the formulation, which is determination of the best freeze drying process or the best tableting process. In simpler form, again, what can XRD do for drug analysis? X-ray diffractometers are used for pharmaceutical raw materials, formulation, R&D, and quality control. If I would like to divide my sample, I would call them, the drug samples, I would call them as API, the active, excipient, the bulk, the coating agent, the binder, and the lubricant. Sample could be in the form of powder, tablet, capsule, or liquid. And the application would include polymorph analysis, new structure, quantitative analysis, degree of crystallinity, or the amorphous point, part, the phase ID, microvolume sample, and microspot analysis. Let's go into the investigation of crystal forms. As I said, the physical state of drug could be in various forms, and that also include the possibility of polymorphism, the single molecule, drug compound only, the solvates, salts, co-crystal, and other molecules. Further, these can be further divided into being in crystal form or the amorphous form. We are aware crystal form is the lattice, which is the repetition of the unit, and amorphous is no such lattice repetition, but their properties, physical state, physical properties are very different, and dissolving of them in the body is also different. So it's very important to be aware of their forms, and XRD is one of the great techniques to see that, as we will see in the coming slides. So let's begin with one of the most important things, polymorphism. Here is a carbamazepine anti-epileptic drug. The form 3 is the desired form, the API. However, it can exist in a form called form 1 or form 4. So we all know polymorphism is the ability of the molecule to exist in different states, basically in unit cell and crystal structure. For example, this one has different space group, triclinic, the monoclinics, and different unit cells. But the most important form would be the form 3. These forms can be converted sometimes by heating or different processes. So that's polymorphism. And XRD is very useful in quickly identifying the polymorphic forms. For example, form 3 has a different diffraction pattern compared to form 4 or form 1. It's a quick signature of every molecule in XRD. As long as your unit cell and space groups are different, you will get a diffraction pattern which are different from each other. Investigation of crystal forms target API. We also know that heat and moisture can also change the API. So the moisture, let's see its effect. Could be dehydration or hydration. There is actually a unique hydration number where we know this polymorphic form or the API will exist or will not exist, and how to convert them into each other. Similarly, heat can change the phase or can amorphize a material, or the processes can go through a melting stages. So these can also be studied from XRD. Phase transformation by heat, ab initio structure analysis of small molecules, we'll talk about that, and dehydration under heat and hydration by humidification. So we are all aware of DSC, differential scanning calorimetry, which is an important technique to study these endothermic exothermic reaction. However, DSC alone at times is not enough, because there are times when these endothermic exothermic reactions are either have a very broad or a small peak and are not visible or cannot be easily identified or overlooked. Same way, they can have very close peak. It is very difficult to identify in the case of continuous reaction. Sometimes reactions cannot be identified by DSC. The form created between two reactions is not identified. But the main difference here between an XRD and DSC would be the forms we don't know. We understand that we are going through a melting, endothermic, and exothermic reactions and changes or co-crystallization, recrystallization, but we do not know which forms we are making. So Rigaku has a XRD DSC combined unit in SmartLabs, which is able to study both X-ray diffraction patterns in situ with the changes that a molecule is going through under the treatments of low temperature to high temperature. So you put a reference sample, usually a corundum or aluminum plate, along with your sample, 3 to 10 milligram, and you can go from very much from minus 40 degrees centigrade to 350 degrees centigrade. And you can also provide different atmosphere, static air, inert gas, or humid gas. So let's see an example of a combined XRD DSC in the case of tolbutamide under dry nitrogen. So on the left is the evolution or the ab initio evolution of the XRD pattern, and on the right is the DSC. So there is a phase transformation process. For example, this is time scale, this is temperature scale, the blue lines, and we are seeing a different evolution. So for example, form 1 goes through a phase transformation as indicated by this endothermic reaction around 112, and then it goes to melting at around 129 degrees centigrade, stays in that state, and then an exothermic reaction indicates a crystallization again to an unknown form. So form 2, form 1, and amorphous, and form 4. So these things, how do we know? Because from the simultaneous collection of XRD pattern, we can see the XRD data evolution. We are going from form 2 goes to form 1 in blue, and then there is a melting process. We are essentially in a state of no crystal structure. There's an amorphous form, and then there's a start of crystallization again to an unknown form. Which brings me to my next topic, an unknown form. So XRD can also, diffraction pattern, we can find the brand new structure of a material. Usually, the easier or a better approach is always find a single crystal and know the new structure of a material. But sometimes crystals do not grow big enough, or under the temperature changes condition, they are so small that one cannot collect a single crystal of a suitable form to do a single crystal work. In that case, it is quite common to perform a full brand new structure analysis using a structure solution powder work. This involves preparing a clean sample, a good high resolution and high intensity XRD data. And the powder diffraction pattern, then what we do is indexing, which is identifying the unit cell. We extract the intensities from this then and assign a space group. Then involving one of these methods or a combination of these methods, direct space method, charge flipping methods. Essentially, all of them are energy minimization techniques to find the best structure which fits under these conditions. And then there is a read fill refinement process after also finding the unknown different peaks from the different Fourier maps. And this sort of now 90% found structure is refined, and rest of the molecules are built using read fill refinement, and then you can verify the structure. This is a very involved process, finding a brand new structure from powder diffraction pattern, and Rigaku provides through SmartLab Studio 2 a package dedicated to it. It's called Structure Determination Package, which takes you step by step through all these processes to find a brand new structure. An example is given here of acylcovir form 4 measurement temperature at 210 degrees centigrade using Rigaku's SmartLab in an EU Hansen monochromator. This shows the final read fill refinement where a model has been fit on the actual powder diffraction pattern. So usually the powder pattern is red in color and the model is blue. It is completely masking, and this is the diffraction pattern. Here are the residue, goodness of fit, and you have the brand new molecule found. So in structure determination, I want to point out that one of the very requirements is having a very high resolution and intensity on the peak because we perform indexing. So if there is an overlaps of peak, the indexing cannot be correct, and your very beginning process can be slightly erroneous. So Rigaku's Johansson geometry provides us a K alpha 1 system. For example, this is quartz 5 fingerprint region, and because there are K alpha 2 lines here, and you can see in the K alpha 1 system, we are maintaining good intensity, and we have eliminated K alpha 2 lines. So the peaks are very well resolved, and it would be ideal for indexing. One more thing I wanted to mention, the geometry, K alpha 1 geometry is easily switchable with just the removal of the monochromator. And there is a rotating design that the X-ray generator part moves back, and you can put the K alpha 1 optics. So easily switchable Johansson geometry. Let's move on to now quality check of API powders. So once you have an API powder, it could again be in crystal form or an amorphous form. There can be small amount of polymorphic forms in this, which we want to minimize, obviously. And polymorphic forms in the excipient mixture, the one you are adding to this, can also be present or could be enhanced or reduced. So we should all know this. Where is this leading to? So let's begin step by step on this also. One, you can check whether it is crystalline and amorphous by doing a crystallinity analysis by X-ray diffraction pattern. So essentially, the principle is based on scattering intensity by crystal, integrated intensity of these peaks, the area under the peaks, integrated intensity, and divided by the total, the scattered intensity by crystal and amorphous. This can give you a very quick idea of what is your percent crystallinity. As you see in this example, the amorphous content, or the amorphous hump, we call it, is larger, and hence the crystallinity is lower. So this is easy. There's a program to do that. How about a trace amount of polymorphic forms? So for example, in this case, our desirable form is tolbutamide form 1. However, we could have a trace form, undesirable polymorphic form, and we can know this very quickly. This is Rigaku's MiniFlex. A 1%, 0.5%, 0.2% of this form to undesirable is shown, which could be easily identified in a 1-minute scan. And the lower the better, as I said, so you could easily find out how much the polymorphic form is present. Then we go into some other quantitative phase analysis. One of them is calibration curve, where you collect data on desired peaks in different amounts, and you can extend this calibration curve to know an unknown amount of the curves. This is very well-known method, almost like we can call it an Excel sheet method. We have this calibration curves method. Then you have reference intensity ratio method, where a single peak intensity, which has already been compared with, let's say, corundum, can be compared for each of the phases to know their relative amounts. Rietville, as developed by Dr. Hugo Rietville, is a model-based method where, based on the phases, the computer is able to draw an artificial pattern, which is laid upon the exact pattern, the pattern that has been collected on your diffractometer. And hence, from there, you can know almost every information about your molecule, whether how much each phase is present, what are their bond distance, bond angles, and so on and so forth. And crystal structure parameters are of paramount importance for this. I would also like to spend a few slides on Rigaku's new developed direct derivation method. Dr. Hidayo Toraya has been spearheading this method, where using chemical composition, basically the chemical formula, multiple peak intensities in a wide range 2 theta can be assigned to each phase and can be ratioed. So this method, the idea is it doesn't involve any database entries. You can give the chemical formula to the known phases, and then any mixture of these phases can be quantitatively determined. So if I go into the quantitative analysis in a little detail, the classic calibration curve are good up to 0.1%, but depends on the quality of calibration curve. So once you have 2, 3 known, you can create a calibration curve. And in that calibration curve, you can put your unknown, and it will spit out what is that unknown amount, because it is on the calibration line. The RIR quantification, easy, but low accuracy, because they don't account for any preferred orientation at times, and they are based on single peak integrated intensity. WPPF, including Rietville, whole pattern powder and fitting, uses a model, as I explained, And they are quite good and can go as close as up to 4% in relative amounts of the phases, an error of up to only 4%. So that's a very well-known method nowadays, read pull method. But let's talk about this new method, direct derivation method, developed by Hideo Toroya, and I have listed his publication here. Basically, it says the sum of square of structure factor is proportional to the number of chemical formula units. So by knowing the chemical formula, we know the structure factor, and hence this will help us find the relative amounts of each one of them present there. So total integrated intensity is proportional to the chemical formula. This thing helps to find things without the help of the database. So for example, using the C2 function in this DD method, extracted profiles with background of each phase are fixed and scale factors are refined. We can do that because now we know their chemical formula. So remember, I have been emphasizing the important point here is to know the chemical formula of each of the phases. And then by refining the scale factor, you can know their relative amounts. So why I'm emphasizing this? No time?
No slides. I think the slides have gone. The slides, I can't see the slides.
Slides. I'm told I can't see slides. Let me try again. Yeah, good. Thank you. Sorry again for small interruption. I was told slides are not visible. So I was explaining about the DD method. So as I said, one of the important part lies in the C2 function that I was explaining. For example, if you know the chemical formula of your amorphous part, and you know the chemical formula of your crystalline, and once you have stored them in your computer after collecting the data on each of them, then any mixing of this, especially when the amorphous content is going higher and the crystalline component is going lower, which is what is desired these days, you can tell them without the use of any database. An example is shown here for indomethacin. The crystalline form is only 3%, and it reduces to 0.5%, but could be easily spit out by the program. 0.5% was told as 0.71, 1.09, 1.5, 1.5% prepared resulted in 1.65. And you see in the bracket ESD, which brings the results very close to the expected ones. So I hope this method is clear to everyone, where by using the chemical formula, we can go very low about the amount of crystalline form and a large amount of present in a large amount of amorphous form. So moving to, and I'm sorry, I'm taking a little bit more time. There were some little glitches, but incidentally, things are fine now. Formulation, moving on to formulation, the product candidate. You could finally reach into the formulation from two approaches: freeze-dried product and tablets and capsules. So again, in freeze-dried product, you could use cooling or heating and crystallization and the glass transition temperature in either vacuum condition, which can also result in phase transition. You can go to crystalline form, and again, this is true for cavity and coating. So basically, in the product candidate, you are again going by either freeze-dried method, or in the process of tableting something or making capsule. You could also again have situation where polymorphic form appear or disappear, or the desired API form can change, and this needs to be checked. So two methods I want to point out here. Again, we discussed in detail XRD DSC, which is able to see these changes, especially during freeze drying, -40 to -350 degrees centigrade, as I showed in the slides, and I'll show one more: thermal reaction, crystallization, glass transition. And using our TTK 600 from -190 to 600 degrees centigrade, this is essentially very good for freeze drying method, and also in vacuum, you can see these presence or disappearance of polymorphic forms. So again, very quickly, this is a XRD DSC combination using Rigaku's SmartLab equipments, where you can see the amorphous form from very ice cold temperatures and phase transformation into the desired forms and other forms. Again, The DSC graph is shown on the right side, and XRD evolution of the pattern from, again, going through glass transition temperature and from amorphous to crystalline is shown here. I can essentially go slightly faster now to catch up. So in formulation, there is then measurement after milling. One simple way is Bragg-Brentano, reflection geometry, crystal form change, and crystalline amorphous form can be identified. And a special, and even on a tablet, you can perform. But let's kind of moving to the next slide. Only surface of the crystal contribute to diffraction in case of reflection geometry. But we understand sometime we need to be aware what is inside. So we do provide a transmission method, in which case, as I show in this picture, there is an outer surface, then there's slightly inner surface, and then there is more inner surface. If you want to see that, all the gadgets are available to perform such a transmission XRD data collection. Bells and whistles of that are shown here. So this is from the surface, as you can see, and identified as some phases. Inside, you can see a different set of phases, and deeper inside, you see an API. So the bottom line, again, is to show that different phases at different depths can be easily seen with this transmission geometry. So Rigaku solution for pharmaceuticals. MiniFlex, one of our benchtop equipments, along with other surface larger equipments, is very capable of performing a number of these operations. It has low weight, internal and external chiller, high power, 600 watts, variable knife edge or scattering protector. I welcome you to join our workshop and see that. Variable and fixed slits. It can come also a 1D detector or 2D high-pix detector, sample changer ASC or 6 or 8, non-ambient chambers, airtight sample holder, transmission and reflection holders, liquid sample holder, and again, electronic record and electronic signature mode, CFR Part 11 compliance. It could even be put inside a glove box as shown here if required. One of our customers, several of our customers have actually put it in this fashion. So it's shown here. Rigaku's SmartLab SE also provide a number of solutions: automatic component recognition, automatic alignment, state-of-the-art 1D, 2D detectors, range of patented CBO, cross-beam incident optics modules, range of non-ambient chamber, including unique Reactor X and XRD DSC, pre-aligned capillary attachment and automatic positioning of the beam. Again, they also come with 6, 10, or 48 position sample changers, and automated transmission and reflection sample changer. We did talk about Johansson K-alpha-1 geometry for this, and there is also a number of detector available for this. So I won't do a full justice if I just speak one slide, because there's a number of SmartLab equipments available which can also perform SAC, small angle X-ray scattering, DSC. These are possible using these attachment and even large sample changers such as ASC48 sample changer. Let's see some of these geometries now for the SmartLab. So we have divergent and parallel beam. There are situations, as I explained, where you would need, especially for transmission mode, a convergent beam, which is provided with this specially designed cross beam optics, which could convert the beam with just a switch of slit from BB to PB, Bragg-Brentano to parallel beam, or CBOE elliptical, BB to convergent beam. Very good for transmission cases, capillary data. And for each one of them, I'm also showing here that resolution is excellent on this compound, and especially convergent beam for film transmission provide excellent resolution on the peaks using the same material, so that we are comparing them apple to apple scenario. So excellent resolution, full width at half maximum, using different kind of desired beam for a specific sample type. You can also do high throughput polymorph screening through AC6 or AC10, these auto sample changes where you put the samples in all these 10 and remotely control it, and the data would be collected. You can extend it to 10 sample or 48 in reflection transmission sample changes. High throughput polymorphic screening is also possible using these disposable 96 well plate compatible sample holder, high flux X-ray sources available for that, rotating anode, transmission and reflection, both capabilities exist. And again, these are CFR 21 Part 11 compliances, and multifunctionality such as XRD, DSC, and humidity options are available. We have airtight sample holder for samples which are either toxic or air sensitive. This is also liquid sample holder. One such is shown, sunscreen lotion, and you can see that there are different forms present in this liquid that have been easily identified. Qualitative analysis of components contained in 2 types of sunscreen lotion are shown in this picture. So liquid samples are easily, sample holders are there for running such samples. The special case of low quantities, in case of MiniFlex, I want to show a 0.05 milligram theophylline in our low background or zero background, whichever way. This is silicon 510 cut sample holder, and you can find theophylline anhydrate even at this low amount. So basically, if you can see a sample, you can collect a data on it. And a 2-minute measurement scan on bronchodilator theophylline is also shown here for a very low mass percent, 0.1% is shown here. Now, you can collect all these data. How about analyzing them efficiently, quickly? You can have a high throughput polymorph screening through this cluster type analysis where this color from principal components are divided into different colors, and so that you know these patterns belong to class 1 by color, class 2 by color, or class 3 by color. And using this Dendrimer diagram, you can very well analyze them, and you don't have to spend a lot of time on each pattern. Rather, you have groups of these groups of pattern based on principal component analysis, which can be analyzed. SmartLab Studio 2, the software, is integrated X-ray diffraction software package for both measurement analysis now. So everything is included in one package. You can perform analysis while the data collection is going on. User privilege management, you have flexible user rights for administrator operator. So you can do the audit trail, which we have said several times. ER and ES mode are possible. SQL database data can be transformed to. Audit trail viewer, network and site licenses, report creation tool, multiple data processing and visualization tools are present, synchronization with external equipment, and user-friendly flow bar for navigation. 1 minute only. So you can have texture effect in materials, and this can be minimized using the transmission geometry as shown in this slide. So there are several sample holders available to minimize the texture effect. Just wanted to say that. I'm going to come to summary. So XRPD finds application in identification, quantification, and characterization of compounds in R&D process and quality check of pharmaceuticals. XRD DSC attachment, as I showed you, enables identification of crystal forms and study of freeze-drying process, and Rigaku offers versatile tool for pharma research, including high-end multi-purpose X-ray diffractometers, as well as the advanced software SmartLab Studio 2 with ERS mode and audit trail. I would like to thank my colleague, Message Griswa, I hope I pronounced his name correctly, for sharing a lot of materials with me. Thank you very much, Doctor. Thank you very much for your kind attention, bearing with this live presentation. Apologies for some little hiccups, but I think we reached the conclusion slide. Thank you very much again.